Hi, and thank you for joining us today. My name is Matthew McGann. I'm the Marketing Manager here at RedShiftBio, and today I'd like to introduce you to the AQS³ Pro and Microfluidic Modulation Spectroscopy. The AQS³ Pro is a new tool for the biophysical characterization of biologic drug products. RedShiftBio is a venture-backed innovative biotechnology startup who has gone through multiple rounds of funding and is now backed by two of the largest life science instrumentation companies. We have developed a tool along with the software required and expertise needed to deploy this system into the biotherapeutic development pipeline. We have an experienced management team with track records of success in both large instrumentation companies and commercializing disruptive technologies. This platform is the AQS³ Pro. It's powered by Microfluidic Modulation Spectroscopy and represents the next generation in protein structure determination. It really does offer a novel tool that can strengthen your bioanalytical toolkit and provide critical insights to aid in decision making. So what is it that MMS can do for your organization and your research? Well, using automation, it reduces time and therefore cost of development. It can improve product quality by detecting previously unmeasurable changes in protein structure, which allows you to de-risk your product from downstream failures. And consequently, the data quality allows you to make confident decision-making to really accelerate your development timelines to get your drug product to market sooner. MMS is validated by its rapid adoption in the industry since 2019. We have a number of commercial partners, including Amgen and Janssen. We've worked with universities such as the University of Washington and the University of Delaware, and we've also been published in a number of key journals to support the use of this tool in the biopharmaceutical development pipeline. When looking at the tools used in research and development, there are four key attributes that are generally considered during the purchasing process. And these attributes include, is this instrument going to contribute positively to development time? i.e., is it going to reduce the amount of bench time, or is it going to allow for faster decisions to be made? Does the system provide automation? Does that automation provide benefits of reducing hands-on time in the lab? And does it improve data quality by minimizing handling error? Is the information quality provided by that tool precise and accurate enough to be used in the decision-making process? And finally, is the data that it's giving you relevant to the decision that you have to make about your drug candidates? So today I'd like to have a closer look at some of the properties that need to be understood of a biologic drug product and also a drug substance. In terms of a drug product, there's a whole range of factors that need to be measured, and most of these will have some kind of impact on the efficacy or safety of the product. So these properties can include the protein content and its concentration, the breakdown products, and even impurities that could have been introduced through the manufacturing process. So let's move a little deeper and look at the biologic drug substance, which is a subset of the biologic drug product. The drug substance for biologics is generally a proteinaceous material, and so we need to characterize it around a range of both biophysical and biochemical characteristics. These characteristics are recommended by regulatory bodies and USP pharmacopoeias all around the world, so there's almost an unlimited number of characteristics that can be measured. But there is one consistent thing: regulatory bodies are always asking for more information. So the general basic content that we like to measure is things like molecular weight, aggregation profile, melting point to look at the stability of the material. But one of the most important characteristics is structure. Is the protein folded and in its correct and active state to be effective when it gets to a patient? So I'd like to look at protein structure in a little more detail. So when measuring protein structure, we all know there's 4 different types of protein structure: the primary, secondary, tertiary, and quaternary. And there's an almost unlimited number of tools available to us to observe the properties of these different facets of protein structure. But not all tools meet the requirements of the biopharmaceutical industry. And that is really that it has to be a robust, automated, and high quality instrument that produces data that is really relevant and fit for purpose. So let's look at each one of these structural components individually. We're talking about primary structure. We've certainly got techniques like mass spec available to us that definitely meet those requirements of being automated and high quality, and it definitely produces fit for purpose data. Tertiary measurements, including instrumentation such as NMR or hydrogen deuterium exchange, are now really starting to meet the requirements. NMR has certainly increased the molecular weight that is observable, and HDX is really being integrated into the industry at a rapid pace right now. Quaternary structure, there's a number of traditional techniques that have really always met the needs of the industry. Size exclusion chromatography is probably the most incumbent, and dynamic light scattering for looking at quaternary structure and aggregation is also one of the most incumbent techniques. But let's go back and look at secondary structure. The technologies that we have available to us, typically circular dichroism or FTIR would be the first 2 that come to mind. Neither of these tools are really fit for purpose. They do not have automation, they're generally not high throughput, and generally speaking, they may not even be compatible with a formulated protein sample. So let's have a look at these in a little bit more detail. Secondary structure really is a fundamental and important part of assessing the quality of a protein or biologic drug. The existing technologies, as I mentioned, really don't meet the needs of the industry at the moment. FTIR can be quite an onerous workflow if you're using a single ATR cell where you have to clean and replace the cell between sample and buffer measurements. And then even the buffer subtraction processes can lead to quite difficult processing challenges. In terms of CD, the concentration range that the system works in is quite low, and we know that most therapeutics are now moving up to higher concentrations. And CD has little compatibility with buffers and excipients, meaning that you can't measure secondary structure of a material in the format you need to, which is the formulated substance. But why is secondary structure important? There is a direct relationship between structure and activity. Secondary structure directly measures conformational change if you're looking at things like ligand binding or protein-protein interactions. It provides a direct handle on stability if you're doing things like formulation development, manufacturability, comparability type assessments. Even aggregation can be measured and understood in a more deeper level using secondary structure by understanding the composition of things like parallel versus anti-parallel beta sheet, which is a direct indication of the presence of an aggregate in a sample. Finally, looking at secondary structure as a fingerprint tool, you could use it for biosimilarity comparisons and even quality control comparisons for looking at batch-to-batch reproducibility. So secondary structure, although it has been relatively ignored by the industry because of its challenges, really does actually provide and meet the needs of the industry as a measurement that is essential through the characterization process. The AQS³ Pro, utilizing microfluidic modulation spectroscopy, is a secondary structure assessment tool that does meet the needs of the pharmaceutical industry. It measures secondary structure of a protein in formulation conditions, allowing you to obtain relevant results sooner. It provides high quality data, which makes decision-making easier. The advanced analytics provides quality fit for purpose data that is correct for the decision that you need to make. And finally, it is a fully automated system, reducing hands-on time and the errors associated with sample handling. So what does MMS actually measure? Its unique combination of laser and microfluidics really allow it to overcome many of the challenges from the current instrumentation set. And its microfluidic cell is what really has the power. The microfluidic cell has two flow paths which allow it to rapidly alternate or modulate the buffer and sample through the flow cell and observation zone. This means that the system can directly subtract the buffer signal from the sample signal, giving us a differential absorbance spectrum. These data points are interpolated to form the spectrum. The spectrum is then converted into a second derivative, which is a traditional way of amplifying differences between spectra. The second derivative can then be flipped and baselined to give us a similarity spectra. This is the spectrum that we would use to compare and observe the percentage similarity of one spectrum to another based on the area under the curve. Next, this spectrum could be deconvoluted using traditional deconvolution methods. This is a normal Gaussian deconvolution, and the secondary structural elements can be taken out of that, combined and generated a plot or a distribution of higher order structure content, where it can give you information about the percentage of alpha helix, beta sheet, and so forth. So now that we understand a little bit about how the instrument works and the background of the area of measuring secondary structure, I'd like to run through a couple of application examples that we've developed with industry leaders. So when it comes to new and emerging tools in the biopharmaceutical industry, I find it easier to discuss how these tools can be integrated into typical workflows that are traditionally used for characterizing biotherapeutics. So this workflow is an aggregation workflow, which is a workflow performed on every biotherapeutic, and it's designed to assess the presence and quantify the number of particles across a wide particle size range from nanometers up to micron size particles. And what you can see here is I've got 5 particle sizing techniques. All these techniques tell us that the particles are there and can tell us their size or their number or some derivation of that. But in an aggregation workflow, there's another level of information that can be obtained to help that workflow. And what that is looking for the presence of aggregates through structural change. Microfluidic modulation spectroscopy has the ability to identify anti-parallel beta sheet. Now this is an indicator of the presence of an aggregate, and the sensitivity of MMS means that you can see that aggregate well before something like SEC or AUC would be able to find it, even earlier than what dynamic light scattering would be able to see it. So as a tool in the aggregation workflow, although it doesn't tell you the size of the aggregates, it definitely can tell you if they're there much earlier than any other technique. So let's have a look at our first case study, which is examining the thermal and pH denaturation of a formulated drug substance. This work was done in conjunction with Immunogen. And what we're trying to do is observe the effects of pH and other types of stresses on an antibody drug conjugate. So this work started off by assessing the comparison of the antibody pre and post conjugation, and that data is not shown here, but I can tell you that the comparison of the antibody secondary structure before and after the conjugation reaction showed greater than 99% similarity. So we could assume from that that the antibody is not affected by the conjugation reaction and the chemistry involved. So we moved on from there and looked at how these antibody drug conjugates respond to temperature. We have 2 temperature environments that were used. The one on the left, 50 degrees, you can see the size exclusion profile clearly shows the presence of higher molecular weight species in the thermally exposed sample. However, if you compare the higher order structure composition, so that alpha helix beta sheet composition, you can see that structurally the sample is actually not changing. So rather than saying that these materials that are higher molecular weight are aggregates, they're more correctly can be described as self-associated particles. Aggregate generally has an inference that there's some kind of structural change that has occurred, and in this case it appears that there has not been. Now, the benefit of this is that particle-particle interactions can be controlled by improving the formulation. So it may be possible to prevent those aggregates simply by changing the formulation conditions of that particular ADC formulation. The temperature on the right-hand side of the screen, the 70-degree experiment, you can see the size exclusion profile again clearly shows a large amount of aggregate, and in this case it's almost all of the sample that is in the higher molecular weight species. Now if we look at the spectra, you can clearly see, look at the results of the spectra, you can clearly see that the higher order structure has changed. And we can show that the parallel beta sheet has started to form anti-parallel beta sheet. And we know the formation of anti-parallel or intramolecular anti-parallel beta sheet is one of the best indicators of the presence of unordered aggregates in a sample. So this is a clear indication of the strength of MMS, that although this is a very large change, by detecting very small changes in the presence of anti-parallel beta sheet, it may give you a handle on aggregation or aggregates being present before other techniques may be able to. So here is an extension of the same data set. This extension was a sample that had been placed at a pH 3 hold for several days. Now, as you can imagine, this protein didn't like pH 3. So you can see again here, relatively speaking, the chromatogram shows that the stressed material has a large portion of high molecular weight species. If you compare the two spectra in the top left corner, the second derivatives are very different to one another. And you can see that around 1639, there is a great difference, which represents the great differences that we're seeing down in the bottom in the structural plot, where the amount of parallel and anti-parallel beta sheet is considerably changed, but the amount of other spectral features like turn and alpha really aren't changing drastically. So this also supports the argument that this type of spectral analysis allows you to observe the formation of aggregates by looking at the presence of or absence of anti-parallel beta sheets. So here is another very typical workflow used in biotherapeutic development, which is aimed at developing and optimizing a formulation for a given protein. When you're looking at formulation, what you tend to need to do is to look at a range of properties that measure both the particle-particle interactions, the range of charges that are present in a sample, what effect some of the post-translational modifications are having on the global formulation, and even look at characteristics of stability like the molecular weight profile, aggregation profile, and sometimes even the thermal properties if you're looking at melting experiments and so forth. So how does MMS integrate into this type of formulation workflow? Well, secondary structure can give you a handle that is complementary to almost all of the questions that are being asked in this workflow. Higher order structure or even secondary structure could be used to complement things like the aggregation for chemical stability, the colloidal interactions to see if you have any particle-particle interactions. These are generally shown in the beta sheet area. Even the post-translational modifications can affect the secondary structure. So having a handle that actually shows that the structure of the protein is either not being affected by a formulation condition or is being changed, and how that change is occurring in the formulation conditions will allow you to choose the most optimal formulation. So secondary structure really is an important facet of formulation development that typically has actually not been used because secondary structure is so difficult to measure in these types of formulation environments. And the AQS³ Pro really does give you that benefit of being able to measure secondary structure in very complex environments like complex formulations. So as an example of the application of MMS to formulation development, we actually did a project with Amgen. And in this project, we were looking at the effect of polysorbate 80 concentration on secondary structure. So to examine the effect of polysorbate on a formulation, Amgen took a 5 mg per ml antibody solution and added varying concentrations of the polysorbate from 0.01 to 0.1% weight per volume. Now, if you look at the spectra on the screen, you can see here that the spectra have good high reproducibility. In fact, you can barely see that there is more than one spectrum there. So let's look at the numbers to really understand the story. Now you can see the percent similarity of the replicates shown here are all greater than 99%. And if you then look at the higher order structure to see if there's any changes there, you can actually see that all of the higher order structural components have very good similarity as well, with beta sheet being about 61%, turn about 29%, and so forth. So these high similarity scores and the consistency of higher order structure really says that polysorbate is not affecting the secondary structure of this antibody. So this would support the use of polysorbate as a formulation excipient. So the second part of this research, Amgen wanted to look at the effect of protein concentration. So they took the sample both in formulation buffer and in phosphate buffer, and they made samples that ranged in concentration from 80 mg per ml down to 1 mg per ml. And what you can see here is that the similarity of these, when referenced against the 5 mg per ml formulation buffer, all shows greater than 98% similarity, which in this case indicates that concentration also does not affect secondary structure. So MMS really is an essential part of your biophysical toolkit. Although it directly measures secondary structure, the range of data that it provides is highly complementary to a number of other systems that are used to characterize biopharmaceuticals. Things like the presence of beta sheet, which if you are looking at aggregates through dynamic light scattering or size exclusion, is a great example. You can also use the secondary structure through a thermal experiment to complement differential scanning calorimetry, or utilizing the changes in secondary structure in a binding event may complement an experiment done using hydrogen deuterium exchange. So secondary structure in and of itself is a great measure, but it provides information that is highly useful for validating some of the other techniques that are really traditional in this toolkit. So let's wrap this up and talk a little bit about the AQS³ Pro instrument and how it actually does its measurements. We believe that there are 4 key features to this instrument which provide its significant benefits. A high power quantum cascade laser, which gives you about a 30 times improvement in sensitivity. A high precision microfluidic cell, which is what enables both the concentration ranges and the reproducibility that we can achieve from spectrum to spectrum. An advanced analytics package and easy to use software platform, which provides simple data that is usable and the right data that is needed for decision-making processes. And then finally, the automation, which is really the critical part. The fully automated system, which means the instrument can be operated hands-free and we can minimize the risk of human error caused by sample handling. So let's look at each one of these individually. The AQS³ Pro utilizes a quantum cascade laser, which operates in the wave number range of 1590 to 1710. This wave number range is actually the amide 1 band of a protein and allows us to probe the conformational changes of the backbone and hydrogen bonding changes as proteins undergo structural change. In combination with microfluidics, the system is able to produce results with minimal laser drift and very few baseline issues that you would find in other techniques like FTIR. The automated background correction, which is facilitated by the modulation of the sample in the flow cell, also improves background referencing and significantly improves the sensitivity of the technique. So let's wrap this up and talk a little bit about the AQS³ Pro instrument and how it actually does its measurements. We believe that there are 4 key features to this instrument which provide its significant benefits. A high power quantum cascade laser, which gives you about a 30 times improvement in sensitivity. a high-precision microfluidic cell, which is what enables both the concentration ranges and the reproducibility that we can achieve from spectrum to spectrum. An advanced analytics package and easy-to-use software platform, which provides simple data that is usable and the right data that is needed for decision-making processes. And then finally, the automation, which is really the critical part. The fully automated system, which means the instrument can be operated hands-free and we can minimize the risk of human error caused by sample handling. So let's look at each one of these individually. The unique microfluidics, which are represented by the cartoon on the screen, is what provides some of the significant benefits in this tool. The Y-shaped cell at the start of an experiment is primed with buffer. And then the sample and buffer are rapidly modulated in the observation point, allowing us to subtract the buffer signal from the sample signal at each alternation. This provides a series of data points which are interpolated to provide the full spectrum. And generally speaking, we expect a reproducibility of greater than 98%. So let's wrap this up and talk a little bit about the AQS³ Pro instrument and how it actually does its measurements. We believe that there are 4 key features to this instrument which provide its significant benefits. A high-power quantum cascade laser, which gives you about a 30 times improvement in sensitivity. A high-precision microfluidic cell, which is what enables both the concentration ranges and the reproducibility that we can achieve from spectrum to spectrum. An advanced analytics package and easy-to-use software platform, which provides simple data that is usable and the right data that is needed for decision-making processes. And then finally, the automation, which is really the critical part. The fully automated system, which means the instrument can be operated hands-free and we can minimize the risk of human error caused by sample handling. So let's look at each one of these individually. The AQS³ Pro is a fully automated instrument, and it includes automation of both sample analysis and cleaning protocols. The instrument is currently compatible with both 24 and now 96 well plates. Samples are presented to the instrument as matched well pairs, meaning a sample and its matched buffer are set next to each other in a well plate. So a 24 well plate can carry up to 9 samples along with some additional cells that have the reference materials, the water and the buffer to allow you to do some of those background subtractions and to validate the instrument prior to measurement. Automation in this case not only reduces hands-on time, but it improves overall data quality by eliminating some of the sample handling errors that can be introduced to measurements and just improving the overall repeatability of sample analysis. So let's wrap this up and talk a little bit about the AQS³ Pro instrument and how it actually does its measurements. We believe that there are 4 key features to this instrument which provide its significant benefits. A high-power quantum cascade laser, which gives you about a 30 times improvement in sensitivity. A high-precision microfluidic cell, which is what enables both the concentration ranges and the reproducibility that we can achieve from spectrum to spectrum. An advanced analytics package and easy-to-use software platform, which provides simple data that is usable and the right data that is needed for decision-making processes. And then finally, the automation, which is really the critical part. The fully automated system, which means the instrument can be operated hands-free and we can minimize the risk of human error caused by sample handling. So let's look at each one of these individually. AQS³ Delta is the analytics software package provided with the instrument, and it is an easy-to-use tool for analyzing the spectra. If we look at this example, we took an antibody fragment and we incubated it from 24 to 80 degrees. Now, looking at the lower workflow here, you can see that the spectra can be converted into a concentration measurement. This concentration is not obtained using the Beer-Lambert law, so it does provide a nice complementary measure of concentration to the traditional techniques. The next workflow, which moves to the upper part of the screen, converts the raw spectrum into the absolute absorbance. The absolute absorbance spectra can be taken through one of two workflows. The upper workflow shown here converts it into a second derivative. The second derivative is then converted to a difference spectra, which highlights the wave numbers where the most significant difference occurs. You can then select these wave numbers and produce a stability plot or trend plot. In this case, it's a thermal trend plot because it is a thermal experiment, which allows you to look at the melting behavior of the protein. And what we can see here is that the data shows that the sample is melting about 70 degrees, and the most significant difference that's occurring is found in the beta sheet and anti-parallel beta sheet composition. Alternatively, you could work through the lower workflow where the second derivative is converted to an area of overlap plot. These area of overlap plots are what we use to calculate the similarity of each spectra. You can then take these and process them using a Gaussian curve fitting model to produce a higher order structure plot. This allows us to look at the percentage composition of things like alpha helix and beta sheet and compare how that composition changes across the data set. You can see here that blue represents the cooler temperatures and red the warmer. And then in this case, the data is showing that the beta sheet composition and anti-parallel beta sheet, again, are where the most significant difference is occurring, with conversion of beta sheet to anti-parallel beta sheet as temperature is increased. So let's wrap this up and talk a little bit about the AQS³ Pro instrument and how it actually does its measurements. We believe that there are 4 key features to this instrument which provide its significant benefits. A high power quantum cascade laser, which gives you about a 30 times improvement in sensitivity. A high precision microfluidic cell, which is what enables both the concentration ranges and the reproducibility that we can achieve from spectrum to spectrum. An advanced analytics package, an easy to use software platform, which provides simple data that is usable and the right data that is needed for decision-making processes. And then finally, the automation, which is really the critical part. The fully automated system, which means the instrument can be operated hands-free and we can minimize the risk of human error caused by sample handling. So let's look at each one of these individually. So let's conclude today's discussion by relating back to one of the earlier slides that I showed, which is the 4 key requirements for instrumentation in the biopharmaceutical industry. MMS really addresses these requirements. MMS can reduce development time by observing change much earlier in the process than most traditional assays used today. The automation of this system provides measurement in both 24 and 96 well plates, reducing hands-on time, but also improving the repeatability of results. This repeatability and reproducibility, which is typically greater than 98% from spectrum to spectrum, really provides the sensitivity to observe even the smallest changes in secondary structure, allowing you to make decisions earlier that may not have been observable. So the data provided by the analysis package, the AQS³ Delta, really provides an accessible and reliable data set that meets the needs of today's pharmaceutical industry. So that brings today's session to a close. I thank you for your attention. We will now move into the question and answer session. So if you do have a question about anything that we've discussed today, or perhaps something that you would like to see, please post that in the chat panel, which is on the right-hand side of your screen. We'll give you a few moments now to go ahead and type those questions, and then we'll go through and answer all of them for you.